Hi, everyone. My name is Yue, and I'm super honored to be able to present my project here today. Hypertension is a very common word in our daily life. Nearly half of the American adults have hypertension. Unfortunately, 25% of those hypertensive patients are drug resistant, which means they do not respond well by taking optimal or maximum tolerated doses of 3 or even more antihypertensive drugs. Therefore, they are highly likely to suffer from brain stroke, vision loss, heart attack, and kidney failure. So how to effectively treat those drug-resistant patients is a main focus of my study. Since traditional drugs won't work, we need to work on new methods, right? There is an important mechanism in our body, which is called central control of blood pressure. Question here, what is central control? I have an easy example for you to understand. When we are experiencing high blood pressure, our heart sends a signal up to the brain and tells the brain that we need to do something to lower the blood pressure. After receiving and analyzing this signal, brain takes certain actions. It activates receptors on blood vessels and heart. It controls the hormone release in adrenal gland and the kidney. All these work together to lower the blood pressure. This is how the central control works. However, till now, the exact mechanism by which the brain takes actions is still not fully understood. Interestingly, in our study, we found that chronic infusion of apelin-13 in the brain of rats induced typical characteristics of hypertensive patients in rats, such as elevated blood pressure, elevated plasma norepinephrine level, as well as enlargement of cardiac muscle. We then set out to investigate the mechanism behind it. Surprisingly, we found that the expression level of one microRNA gene, which is microR21, was drastically elevated, nearly 30,000 times higher after apelin infusion. And this result is consistent with the recent clinical studies that patients with hypertension display high levels of microR21. So theoretically, cutout of this gene in hypertensive rats will produce antihypertensive benefits. And this is a study we're going to confirm in the future. To recap, apelin plays a significant role in the chronic control of blood pressure, possibly through microR21 mediated gene regulation. And we are confident that our research and our result will provide a potent therapeutic for drug-resistant hypertension. Thank you.